And finally, I'd like to introduce the next speaker, which, bear with me just one second. Yes, he will be our last speaker for today. Christopher M. Coley, Marketing Development Manager of Digital Pathology at Panasonic Corporation. He will be joining us with a talk from 1 week to 1 day. Take control of your research by automating tissue microarrays, multiplexing, whole slide scanning, and AI analysis for enhanced bioinformatics. Christopher, over to you.
So basically what we're looking here is a lot of these processes that you can see in the laboratory when you're looking to do some of these research, so they're very manual. And it starts with making tissue block microarrays. And in order to do a comparison, what we've done is we did 100 tissue cores, and we did them in an automated, and we compared that to a manual process. And we were able to program the instrumentation to do this in an automatic way, set up the distancing, where the cores go, keep up with the data that goes along with those cores, the position from the donor blocks that as they come across, and be able to later go back and pull of that information. It also puts them in a very precise depth level, depending upon how many sections have been taken from the donor material block, so that all of the cores are positioned in the same way. And if you do this in a manual method, it's very tricky, and you have to have a good skill set in order to be able to do this. It's extremely time consuming, so automating this is helpful. We've also taken that automated into doing an automated multiplex staining for immunofluorescence. Instead of having to do a manual method, then scanning those images, those slide images, and then taking the end of those results and doing algorithms with our AI partner to count the cells and be able to do a precise data return. And as you can see here, the equipment from left to right is the 3D Histex Tissue Microarray Master 2, which is a small, great automated device that we'll showcase. In the middle is the LunaFloor LabSat. And then on the right-hand side, we're using the 3D Histech MIDI 2 12-slide fluorescent and bright field scanner. This one in particular uses a Lumicore spectral light source. So automated wise, starting from left to right, we're able to achieve more than 450 cores in a single block. These are used using enhanced punches and drills from 0.6 millimeter, 1 millimeter, 1.5, and 2 millimeter choices. And again, you can establish where on the block you want these cores to go. And you have the option to go in and do very precise coring, as you'll see in upcoming slides, to select the precise cells that you want cored out of the paraffin blocks. So as it says, the TMA Master 2 does have a small footprint for valuable lab space. It's an easy customizable layout in the software that you can see with the diameter shown, and you are able to, over the paraffin block itself, overlay the image of a scanned slide that matches, whether it's immunofluorescence or it's chromogenic IHC or the H&E, and designate very precisely the groups of cells of viability that you want. So if there's areas that have perhaps necrosis, or they have fibrosis or other features that you don't want in your sample set for the cores, you can choose the most precise viable cells with the best staining for it. It does read the barcodes and the faces and take images of those that go into the spreadsheet and the datas to be culminated later, so that you can go back, and as you go through the cores and break them down for the sample staining, it will show you which blocks it came from and which positions in the tissue it came from. So you can see here in the slide layout, I can completely compose my grid. It doesn't have to be as complex. This is an example. We do have several customers with up to 500 cores in a block. You can choose the layouts left to right. You can choose the distance between the cores. You can choose the depth of insertion into the block. All of this is an automated approach that saves a tremendous amount of time over doing the manual process. Here you can see the slide image overlay, which is a really unique feature as opposed to some of the other systems which can be semi-automated. This is phenomenal. Instead of having to try to eyeball, if you have a block that appears homogeneous and you cannot tell the difference of one tissue area from the next, let's say it's just a block of liver, for example, you can take samples from anywhere in there. But if you have a block where there's multiple tissue types in the paraffin embedded sample, this allows you to overlay the H&E or the IHC select those particular cores, and the circle will match the size of the cores that are going to be taken. Select very precise region of interest and insert those in the area on the recipient block that you want. You're also able to take those cores and put them into a PCR tube for molecular testing down the road if you don't want to put them into a block. Again, this is good data tracing to go into the reports. It does capture the face. If your donor material blocks don't have a barcode or a printed material and it's just a handwritten, it still captures that. And if it scans that block again, no matter what, if it's barcoded, printed, or handwritten, it'll go back through and say, I think I've scanned this block before, show you the previous scan, and be able to allow you to say, yes, I've taken cores from this block before. So from a data saving export standpoint, it's phenomenal in being able to keep up with which cores taken from the donor blocks go into which recipient block positions. This is part of a family, right? So the Tma Master 2 holds one recipient block and up to 4 donor blocks. If you're looking to just make more generic type samples, you can change that configuration. You have 5 positions to do it as you please. It does a phenomenal job of drilling into the holes. You can set this up and count down, leave it for overnight if you have very complex. If the 5 block system is not good for workflow and you're making something much more complex like some of our pharmaceutical companies use, the item on the left is our Tma Grandmaster, and it's a real workhorse for people that are doing 400 plus cores in a particular recipient block and want to make lots of recipient blocks. It'll hold 72 blocks total. It uses a drum that rotates for that. It'll have 12 recipient block positions, and it'll have room for 60 donor block materials. So very complex if you want to do very specific arrays and tumor array panels, and that saves you from having to load and change out the blocks in the other workflow. The Tma Master 2 is really good for a lot of our clinical customers that maybe want to make disease panel state blocks for maybe a breast cancer or something along those lines, or for special stains where you really only need a handful of blocks for donor material and nothing as quite complex as you would see with the Tma Grandmaster. The Tma transitions over to the multiplex staining. So once you have that multiplex block array ready to go, you can deparaffinize, go onto the machine, which is the Lunaphore LabSat. So this is a phenomenal way of time saving. These multiplex traditionally can take days to produce and require an awful lot of time of checking, rinsing, going back and forth and through here. And this simply allows you to load one slide at a time and do the multiplex staining and allow the end user in the laboratory to move on to other tasks while this is automated. So this is designed for the chromogenic formalin fixed paraffin embedded IHC, frozen section IHC, and then the fluorescent stains from multiplex traditional IF, frozen section IF. So it's a real workhorse for these types of tools, whether you're doing a quick check on something off of a cryostat, such as a muscle biopsy or renal biopsy in a transplantation case, or if you want to go on to doing a multiplexed off of a formalin fixed paraffin embedded. So these have lots of applications to be used. It's very rapid because it uses microfluidics and pressure in order to increase the time savings that you would see off of a manual type system. It's extremely precise in the fluid reagent management and uses a small amount of reagents when you're going through the system. And you can tweak the customized protocols for optimized staining for various multiplex kits that are on the market today. So again, the microfluidics and rapid is the real key here that you're looking for. These biomarkers in development allow you to have precision control. So if you're developing an assay, you're able to do this quite quickly. You're able to save precious reagents or antibodies if you're developing your own products, and allows you to have that highly reliable reproducibility and control to bring these types of stains into your lab. It is a skill to do these types of stains manually. It takes a fair amount of training and time, and the reagents are quite expensive. So this is a really good way of making the most out of the reagents that you have to order for these types of tests. So the ultra rapid turnaround time is why it's so fast. So again, this goes over to the flash fluid exchange. There's the slide that sits into the apparatus. There is a pressure control delivery of the reagents that allows them to go through and gets the interaction with the epitopes and the markers on the slides very well. And then you can control those incubation steps very precisely, as you can see here, to temperatures above 100 degrees without affecting drying or evaporation or inconsistency across the tissue on the slide. This keeps everything at a nice homeostatic environment so that you're able to conduct these things reliably over and over again. And the results speak for themselves. We've got phenomenal results with various multiplex and even single staining on here so that you can see when you go into scanning later on, you're able to go through and get good, consistent, quantifiable results. So the staining intensities will be the same over and over and again. So it doesn't throw off or require additional calibration steps when you're staining to be able to quantify later on, which adds a lot more time down the road. You're able to get all of that with the tissue that fits up underneath the chip. So you've got rapid incubation and a lot of tissue surface area that you can control for that. So the fast fluidic exchange technology, you're able to do this with multiples. You can do this with the TSA kits. You're able to do this with doing sequentially and specific antibody cocktails. So if you're doing biomarker discovery, you're allowed to do a lot of these markers on here with the TMA slides to get a wide burst of array sample stainings or early on and be able to taper that down as you whittle away for the best staining sets and the best reactivity for the tissue that you're going to have. So you're conserving tissue, you're increasing your availability of sample set, and you're decreasing the amount of time that it takes to conduct that TMA block and the reduced amount of time to perform these very lengthy staining protocols. So here's an example. This is formalin fixed paraffin embedded tissue. This is a human tonsil section, and we used a six-plex marker with DAPI with FOX, PD1, CD8, PDL1, CD68, and cytokeratin. And this was a 4-hour, 37-minute protocol that we ran, TSA-based chemistry, with phenomenal staining results across the entire sectional cores. And again, we did this with 100 cores for this particular application, so we could do a side-by-side comparison for our data coming out on the other side here. And the results are extremely good. We're able to get these nice patterns in here because of how the arrays are laid out, and then later go back in and do the quantification steps. So we move on to the multiplex to the whole slide imaging on the MIDI-2 that you can see on the right-hand side with fluorescence application. With the scanning on here, you can see the bar at the bottom to the left and the right. You're able to put flat trays for these aqueous mount slides for immunofluorescence. And there are shields to protect from the light that's in the room so that you're not sacrificing the fluorophores as you're doing the imaging. So this is a great concept. These instruments in this MIDI-2 come in multiple configurations. They're a small footprint for a good laboratory space. You're allowed to select 1 or 2 objectives on these instruments, actually 3 different objectives now, a 10x, 20x, 40x. And with camera adapters, you're able to expound those up to 96x native resolution before you do any digital enhancements based on the cameras and the lenses on the optical pathway down into the sample here. In the light engines that you're on here are specifically designed to be able to pick up based on the different immunofluorescent multiplex panels that are on the market. And there's plenty of space on the turret for 9 filter cubes. And each of those filter cubes are able to hold anywhere between 1 and 4, so single to quad band filters. So you really have a full spectrum of filters to your availability at any given time without having to go in and make manual changes to the instrumentation. You're able to also, once you do your scanning, as it sees here, you can have several images pulled up at one time. You don't have to review one at a time. You're able to pull up the different channels individually, and then as you want to increase them into a single view of it. The slide viewer pairs well with image analysis platforms. We work extremely well with a lot of the established image analysis and those that are our own creating in our partners. So as you see here, it's a very easy click-through menu. You can select the channels that you want up front. pick the filters that you'd like to have in here, and then you can do an automatic or manual exposure. And using the dark field preview, you don't have to go in and focus on all of them. It will go through the spectrum using DAPI, and it will make a correction so that you're not getting overexpression in some and underexpression in others. It tries to make all of the excitations even, so that when you view the image, everything is on the same level of intensity, and you're not muting out anything, and you're not being blinded by autofluorescence or any of that sort of thing. It calibrates those things so you don't have to go back and do it. As you're developing protocols for an antibody cocktail, you're able to go in and change those manual exposure times and then save them, if you wish, as a protocol that you can repeat without having to make additional manual adjustments. So once you've done this with the TMA module, it takes the grid of cores, and instead of having to view them all as one slide, it will take each of the cores and make them a separate image for review. So you're able to take the 400 plus cores that you have in your array. It will take each one as a separate entity for you to evaluate and do the analytics on, so that you're not having to do them on all of the cores at once. It allows you to break them down and keep that data tissue by tissue, core by core from the donor blocks, so that you can see exactly what the tissue types were and how those resulted in your staining. The segmentation samples based on the analysis and measurements are really key here. So you're able to go in and really count and start doing the deep dive into it. And if you need to do an automated rescan because you're changing some parameters on those results, you're able to go in and easily take each of those individual cores as opposed to the entire block and rescan just those sections. So as we go through this, Quant Center is a software that 3D Histic has various features that allow you to do image analysis as you define. You can do based on specific cell types. You can do the entire slide. You can circle specific areas that you want to do and look for very defined specific features as you want to look at morphological changes, staining pattern changes, quantification and scoring of specific cells. So you're able to set each of those up and then run them on here, both bright field and immunofluorescence. So the Histoquan is one of those algorithms that we use, and it allows you to do segmentation and measurement based on the colors of the cell components. So in the fluorescent area, it's very easy to do because I've artificially assigned those various colors of blue, green, yellow, and red on the filters. Select the area that I want to do and go ahead and churn that out, quantified based on the percentage of the sample that is being evaluated or the whole slide, and the specific channels and colors as we see fit. And we're also able to negate the ones that we're not really interested in, that maybe DAPI is just providing a morphological background. You don't have to include that in the sample counts if you don't want to. So it's a very nice customizable tool. And again, these can be saved as protocols. So from whole slide scanning to bioinformatics, we're partnered with Iphoria, which has a great way and unique tools to do artificial intelligence above the image analysis that we described in the Quant Center. So on the research end, we've got the Fourier Hub. This is all a web-based tool. We use Iphoria Create, and you're able to go in and use the staff or your own, after training, go in and take your sample sets from easy to complex and have these protocols developed as you partner with the company to be able to perform that AI supported analysis. It just is extremely customizable based on the layers and the types of cells you're wanting to look at in both bright field and fluorescence. So as you can see here, this is an example where you've got, you can annotate and train the AI based on the level, the identify, quantify, or measure the features of interest. You can pick as many layers as you want to here. You can see multiples. You can have a stroma that's particularly labeled in green, and you want to be able to see what the percentages of that. And then maybe I have an area of interest, a particular tumor type or disease state. And within that, I'm able to also check out different variations of those cells, very specific patterns of those cells, depending upon what disease state I'm looking for, whether it's going to be cribiform or sheets or various different patterns in those tumor cells. And as I add those layers, it adds to the quantification output that I'm able to do. So you can start simple and build on the complexity as your research grows in that particular set of slides. So here's a more complex based one. This is a cloud-based, of course, deep learning algorithm that's being used. You can define the image analysis that you need, create your bespoke AI model, analyze the images in the model. It's extremely easy to use. In this particular instance of a bright field, I'm able to select in here from the brain tissue, the various cell types that I want to do. And then furthermore, go back in and sift for specific abnormalities or styles of those cell types, and very clearly see while I'm setting up the layers, how this is going to look as I go through this. And it's extremely rapid technology. There's no need for any coding or prior knowledge. You basically walk through and select visually the areas that you're wanting to do, and then you're ready to start training on your models and kick it out. So here's a good example here for the unlimited channels. Again, this is a tool that is as complex or as simple as you want as you're going through here. I can select the entire section on the slide. I can go into very specific regions of interest under high magnifications and get the details as specific as possible based on what I'm looking for in my analysis. And that's the nicest thing, is this isn't a tool that's preset. This is a tool that's important to what you're looking for in each slide in each set of research. So with the this particular one, we did the 6 fluorescent channels for the multiplex. And as you can see here, we're able to simply go in and define what each of those are, what types of cells we're looking for. You can see the annotation, the percentages, and there's a whole host of tools and snapshots that I can make in different regions as I want to look at these in different layers. I don't have to look at each of the stains at the same time. I'm able to filter them out, choose it, get a really crisp view of the one channel that I'm looking for and highlight those cells that I really am interested in doing, and then build upon the next layer. So again, this is a huge impact for the research. The automation of these manual processes on a limited amount of footprint space in the laboratory is really important. It's a huge control. You don't, laboratory researchers that may have not been able to do this in the past. So basically it's designed to do an awful lot of control so that you can bring these tests into your laboratory if you've been sending them out prior to that. And it's able to save an awful lot of time and give you an awful lot of consistency, with the end goal of being able to consistently get your data quicker with less time and reagents. And look forward to whatever questions come up.
Thank you very much, Christopher. It was really insightful.